You've been thrown into the year 1347, medieval Europe. Castles, knights, cathedrals, the whole thing. Beautiful, honestly, for about 2 days. On the third day, you notice people coughing blood in the streets, black swellings on their necks, entire families dying in their homes. The Black Death has arrived, and you're the only person in the world who knows how to stop it. Before you do anything, let's understand what disease actually is. Because in 1347, nobody knows. Not the doctors, not the priests, not the scholars, nobody. Most people think disease is caused by bad air, miasma, or an imbalance of the four humors in the body, or divine punishment for sin. The actual cause, invisible living creatures too small to see, hasn't even been imagined yet. Here's what's really happening. Bacteria. Bacteria are single-celled organisms, living things smaller than a grain of sand by a factor of thousands. They're everywhere, in the air, in water, on every surface, on your skin, inside your body. Most of them are harmless or even helpful, but some of them multiply inside you and release toxins that destroy your cells. That's infection. That's disease. The bacteria that causes the plague, Yersinia pestis, enters through a flea bite, travels to your lymph nodes, and multiplies until the nodes swell into black, painful lumps. Then it spreads to your blood, then your lungs. Then you die, usually within a week. In 1347, this bacteria will kill between 75 and 200 million people across Europe, Asia, and North Africa, roughly 1/3 of the entire human population. You know something nobody else knows. You know how to kill bacteria. Here's the thing that will break your brain. The solution was sitting right there the entire time, growing on old bread, floating in the air, covering forgotten food in every kitchen: mold. Certain types of mold produce chemicals that kill bacteria. Not all mold. Some mold is just mold. But one specific type, penicillium, produces a chemical that destroys bacterial cell walls. Without a cell wall, the bacteria falls apart and dies. This chemical is called penicillin, and it's one of the most important discoveries in human history. The crazy part, people in the Middle Ages already noticed that moldy bread sometimes helped infected wounds heal faster. They just had no idea why. They had no concept of bacteria, no concept of antibiotics. They were accidentally using penicillin without understanding what they were doing. You understand what you're doing. Let me tell you the story of how penicillin was actually discovered, because it's important to understand just how close humanity came to missing it entirely. The year is 1928. Alexander Fleming is a Scottish scientist working in a hospital laboratory in London. He's studying bacteria, specifically Staphylococcus, the bacteria that causes infected wounds. He goes on vacation for 2 weeks, leaves his bacterial cultures sitting on the lab bench, doesn't seal them properly, doesn't put them away, just leaves them out in the open air. He comes back. The cultures are contaminated. Mold has grown in several of the dishes. He's about to throw them away, and they're ruined, useless for his research. Then he notices something strange. Around the patches of mold, there are no bacteria. The bacteria colonies stop growing near the mold. There's a clear zone, a circle of nothing, around each patch of mold. The mold is killing the bacteria. Fleming isolates the mold, identifies it as Penicillium notatum, a common mold found on bread and fruit. He grows more of it. He discovers that the liquid the mold produces kills bacteria even when diluted hundreds of times. He names this substance penicillin. It takes another 12 years before penicillin is purified, mass-produced, and used to save lives. By World War II, it's saving thousands of soldiers from infected wounds. By the 1950s, it's routine medicine, all because one scientist came back from vacation and looked at his contaminated dishes before throwing them away. You're in 1347. You're not coming back from vacation. You're deliberately creating what Fleming found by accident. Here's how you grow penicillin, and I mean actually grow it, step by step, with materials available in medieval Europe. You need bread, specifically bread made from wheat or rye, the kind every bakery in Europe produces every single day. You leave the bread in a damp, dark place, a cellar, a covered basket, anywhere with moisture and no direct sunlight. You wait. Mold will start growing within days. Different colors: white, green, blue-green, black. You want the blue-green mold. That's penicillium. Now here's the critical part. Not all blue-green mold is penicillium. Some is aspergillus. Looks similar but doesn't produce penicillin. Some is other species. You need to test it. You take a piece of moldy bread. You soak it in clean water, boiled and cooled water as clean as you can get. The mold releases its chemicals into the water. You strain out the solids. What's left is a liquid containing penicillin, if you pick the right mold. Testing is simple but requires something medieval Europe doesn't have, a way to see bacteria. But you can test indirectly. You take the liquid and apply it to an infected wound, a small wound, a minor infection, not life-threatening. If the infection improves within a day or two, you have the right mold. If nothing happens, wrong mold. Try again. This is exactly how early antibiotic testing worked in the 1940s before modern laboratory techniques. Trial and observation. It's slow, it's imperfect, but it works. Now, how does penicillin actually kill bacteria? Because understanding this is important. Bacteria have a cell wall. It's a rigid outer layer that holds the cell together under pressure. Inside the bacterial cell, there's water, proteins, DNA, all the machinery of life. The pressure inside is higher than outside. Without the cell wall, the cell would burst like an overfilled balloon. Penicillin blocks the construction of new cell wall material. Bacteria are constantly repairing and rebuilding their walls as they grow. When penicillin is present, they can't build new wall sections. The wall develops weak spots. The internal pressure pushes against those weak spots. The wall ruptures. The cell explodes. The bacteria dies. Here's the beautiful part. Human cells don't have cell walls. We have cell membranes, which are flexible and work completely differently. So penicillin kills bacteria without harming human cells. It's a targeted weapon that only destroys the enemy. This is why antibiotics are different from every other medieval medicine. Herbs, bloodletting, prayer. None of those specifically target the cause of infection. Penicillin does. It kills the bacteria directly while leaving your body untouched. But there's a problem. A big one. Dosage. Too little penicillin and the bacteria aren't fully killed. Some survive. Those survivors are the ones that happen to be slightly more resistant to penicillin than the others. They multiply. You've just created a population of bacteria that's harder to kill. Too much penicillin and you still kill the bacteria, but you're wasting precious material that could treat other people. In 1347, you cannot mass produce this. Every batch of moldy bread water is labor-intensive and unpredictable. Fleming struggled with this exact problem. His early penicillin preparations were weak and unstable. It wasn't until Howard Florey and Ernst Boris Chain figured out how to purify and concentrate penicillin in 1940 that it became practical medicine. You don't have their laboratory equipment. No flasks, no controlled temperatures, no chemical purification methods. What you have is bread, water, and observation. The best you can do is this. Standardize your method. Always use the same type of bread. Always soak it for the same amount of time. Always use the same volume of water. Test each batch on minor infections before using it on serious cases. Keep detailed records of what works. It's medieval science. Observation, pattern recognition, gradual refinement, slower than modern methods, but better than nothing. Here's the historical moment that should haunt you. The Black Death kills 75 to 200 million people between 1347 and 1353. Yersinia pestis is a bacteria. Penicillin kills bacteria. If someone in 1347 had deliberately cultivated Penicillium mold, extracted it properly, and understood even vaguely that it helped infected people recover, millions of lives could have been saved. The mold was there, growing on bread in every kitchen. The method of extraction requires no advanced technology, just water and cloth for straining. The testing method is simply applying it to wounds and watching what happens. Everything necessary existed in 1347. The only missing ingredient was knowledge. One person who understood germ theory and antibiotics, dropped into medieval Europe with the explicit goal of producing penicillin, could have stopped the plague. Not completely, not immediately, but enough to save millions. You are that person. So here's what you do. You find a monastery. Monasteries are the centers of medical knowledge in medieval Europe. They have libraries, gardens, infirmaries for treating the sick. You convince the monks that you have a treatment. You show them moldy bread. They think you're insane. Mold is corruption, decay, the opposite of healing. You insist. You demonstrate on one infected patient with their permission. The patient improves. The monks watch. You demonstrate on another. That patient improves too. Word spreads. You teach them your method. Grow the mold, soak it in water, strain it, test it, use it on the infected, record what works. Not everyone survives. Your medieval penicillin is crude, inconsistent, sometimes contaminated, but more people survive than would have without it. Monasteries across Europe start replicating your method. It spreads slowly. Communication is slow, travel is slow, standardization is nearly impossible. But it spreads. The plague still kills millions, but not a third of Europe. Not this time. Centuries later, historians will find records of the monk who grew mold on bread and saved the infected. They'll call it superstition, folk medicine, a lucky accident. They'll have no idea it was deliberate. They'll have no idea someone knew. Fleming found it by accident. You found it on purpose. That's the only difference.